Hi friends, welcome back. Today we are going to see the topic cell structure and function. What are the learning outcomes? What is a cell? Who discovered cell theory? How to differentiate cells? Now let's start with what is a cell. Cell is the basic structural and functional unit of life. What does this mean? It means that cell gives structure to the organism and it does the functions. So what does it mean basic unit of life? It means that cell is the basis for any organism. That is, it is the building blocks of life. So if it is the building blocks of life, then even unicellular or multicellular organisms come from the same cell. So cells do not divide if they're forming the unicellular organisms, while they divide plenty of times while forming the multicellular organisms. So this is how a cell is gonna look. So a cell has a boundary of sorts and it has many structures inside which are called organelles. We shall learn more about them in the chapter. Who discovered? So, who discovered means who is seeing all of this, who is making us understand what is cell and what is inside it. Who actually found all of this? So, first person was Robert Hooke, and in the year of 1665, he observed the structure of cork cells. So cork cells, what he saw under the microscope was looking like this. It was like small compartments, and these compartments reminded him of the lonely saints in the monasteries. So he coined the term cells, which is used till today. In the year 1674, a scientist named Anton von Leeuwenhoek observed the first living cells. So this means that cork cells were dead cells as we know it, and Anton von Leeuwenhoek is the first person to observe the living cells, functions, and everything that it is doing to live. While Robert Hooke, all he observed was dead cells. So the living cells that Anton van Leeuwenhoek observed was protozoa. Protozoa is a type of microbe, as we know it. Then after this, in 1831, Robert Brown discovered the nucleus. So what is a nucleus? So if we take a cell here, in the center we can see a dot of sorts. Just inside the boundary and inside that jelly-like substance called cytoplasm. The dot that we see is the nucleus. And this nucleus was discovered in 1831 by Robert Brown. After this, in 1868, T.H. Huxley described protoplasm as the physical basis of life. So what is protoplasm now? So we take the same cell, we take the cell's boundary, that is the cell membrane. The boundary is called the cell membrane, also called the plasma membrane. So what we can see here is the nucleus, the cytoplasm. So protoplasm is nothing but the combination of the nucleus as well as the cytoplasm. So this is where many of those structures lie, and these structures are very essential for the life of the organism. So this is why protoplasm was termed as the physical basis of life. Now let's see about the cell theory. In the years 1838 and 1839, two scientists, namely Matthias Jacob Sleeden and Theodor Schwann, put forward the cell theory. So we know that there is a cell theory put forward by these two scientists. So what is in the cell theory? In the cell theory, there are three postulates or subdivisions. The first one says that cell is the basic structural and functional unit of life. Then they've stated that all organisms are composed of one or more cells. They have stated that because we know that there are two small divisions in the organisms. So that is unicellular and multicellular. So unicellular has one cell while multicellular has more than one cell. So that explains it all, that organisms can have one and more than one cell. And the last postulate says that all cells arise from pre-existing cells. This postulate arrived from what Rudolf Virchow stated, that is, in Latin, omnis cellula e cellula. This means that all new cells are born or are divided from the pre-existing or old cells. So now we're going to see how cells are divided based on number. They can be divided based on other contexts. So first context is number. So the divisions based on number are very familiar to us, that is unicellular organism and multicellular organism. Unicellular organisms, that as we know, have one cell which can perform all life functions or activities for keeping the unicellular organism alive. Examples include amoeba and bacteria. This is, of course, the diagram of an amoeba, because it is irregular in shape. It does not have a definite shape. The second division is multicellular organisms, which have more than one cell. So in this organism, each cell has its own specific function for the organism to stay alive. So if one cell does not do its function, then the other one, which does the same function, continues doing that function to keep the organism alive. But that is not the case when it comes to the unicellular organism, as it has only one cell. So if that cell dies, the organism dies. So some examples of unicellular organisms include humans, animals, and other organisms, that is birds, trees, like that. Now, we're gonna see cell division based on the presence of nucleus, nuclear membrane. So, where there is a nucleus, there will be a nuclear membrane. If the nucleus is not there, the nuclear membrane also will not be there. So, based on whether there is the nucleus, so there are prokaryotic organisms and eukaryotic organisms. Organisms which have neither the nucleus nor the nuclear membrane. They have only a structure called nucleolus. These organisms are called prokaryotic organisms. So some examples include blue-green algae and bacteria. So this is a representation of a shaped bacteria. This is rod-shaped, so it's called bacillus. Now we're going to see about eukaryotic organisms. Eukaryotic organisms are organisms which have a nucleus and a nuclear membrane. And some examples include amoeba and paramecium. So I said that nucleus is present, then nuclear membrane is automatically present. So that is because the nuclear membrane what it does is the same function that the cell membrane has. So that is like the cell membrane is selectively permeable. Selectively permeable. The same way, the nuclear membrane is also selectively permeable. That is, it allows only some things to enter and some things do not enter and damage the nucleus. So it protects the nucleus from outer damage. So the next, we're going to see the cell division based on shape. So there are different shapes for cells. So how do we know that the cells are having this much different shapes, sizes, whether they have a nucleus or not? Everything is because of the invention of the electron microscope. The electron microscope was invented by Knoll and Ruska. So now let's see some shapes of bacteria. There are four main shapes, that is, and the names according to the shape: bacillus, rod shape; coccus, round shape; spirillum, spiral shape; vibrio, comma shape. Next is eukaryotes. Eukaryotes have columnar, squamous, cuboidal, elongated, biconcave, cylindrical, thread-like, and branched and filamentous. So let's see some examples for each of them. So starting off with some examples of bacillus. So Bacillus subtilis, Coccus streptococcus, Spirillum azospirillum, Vibrio is Vibrio cholerae, then we have the shapes of eukaryotes, that is columnar, squamous, and cuboidal. All are shapes of epithelial tissue about which we are going to learn further in the chapter. And elongated is for muscle fibers. Biconcave, that's an interesting shape. That is, we all know about the concave mirror. That is, it's like this. It has a reflecting surface over here, the same way it's biconcave. Just how we draw a tree, the same way, and this is biconcave. So one example is RBC. So the next is cylindrical. Some algae like Spirogyra are cylindrical. And thread-like are neurons. Branched and filamentous. It's an interesting shape again. That is the shape of rhizopus. That is also called bread mold. Now we're going to see the cell division based on size. Now the sizes of cells are measured in microns. So some examples of the cells and their size is given below. That is RBC is 7. This is micrometer. Micrometer is the same as microns. Both are the same things. Both are used for measuring the sizes of cells. So RBCs, as we saw in the slide before, it is biconcave in shape, and it is 7 micrometers. The next one is ovum. Ovum is 100 micrometers, and a fun fact is that it is the largest cell in the human body. Very interesting. And the next one is brain cells. That is 0.005 micrometers. And a fun fact about this as well, this is the smallest cell in the human body. Now we're going to see what a cell contains. So the 3 most important and broad divisions. So it is the most important and the most broad divisions. So that is like just 3 divisions. That is what we have already saw before. That is cell membrane, cytoplasm, and nucleus. So first we're going to see about the cell membrane, more information. It's the outer covering of the cell, protects the cell from invasions, that is, it does not allow it inside the cell. It is selectively permeable, that is, it does not allow the other germs to infect the cell, so it does not allow it inside. It only allows in food material and some other things that are useful for the cell, not harmful for the cell. Then we have cytoplasm. Cytoplasm is a jelly-like substance that acts like a transport medium, and it has some cytoplasmic organelles, that is the structures that we saw before, that is in the cytoplasm. They are called cytoplasmic organelles. We're going to see about them in the next module. Then we're going to see about nucleus. So nucleus is the control center of the cell, centrally located. That is, if you take a cell, it's right in the middle. So it has chromatin and chromosomes. It's the most important part. So chromatin is thread-like, and this thread-like substance has the genetic information in the form of a very familiar structure. That is in the form of DNA. What is the full form of DNA? The next question. So the full form of DNA is deoxyribose nucleic acid. So that is DNA. Now, so next what we have is chromosomes. So chromosomes are pairs of chromatin. So when we say pairs of chromatin, it is not like this, not at all like that. It is paired in a special way like this. This is a chromosome. So chromosomes are in all the organisms, and they are in specific numbers. In humans, there are 46 chromosomes, that is 23 pairs. So 22 pairs of these 23 pairs are normal body chromosomes, that is the things that will decide how you look, that is the genes, like you have your mother's nose, you have your father's eyes, like that. So it will be hereditary traits will be stored in the chromatin and the chromosomes, because chromatin paired is chromosomes. So these body chromosomes are also called autosomes. And the 23rd pair, it's very important. So let's see about the 23rd pair. So the 23rd pair is called the sex chromosome, or the allosomes. Why is this 23rd pair so important? It is so important because it determines the gender of the baby. So 22 pairs of chromosomes all will be same, but the 23rd pair will have an X chromosome and a Y chromosome. Y chromosome. So if it is an X and an X, it's a girl. If it's an X and a Y, it's a boy. This is why the 23rd pair is so important in determining the gender of the baby. Thanks for watching. If you liked my video, please like and share. Subscribe for more videos.